Our group really enjoyed hearing from Naveen a couple months ago. He shared a lot of great information about Viome, how Viome thinks about microbiome and gut health. He talked to us about transcriptomics and the importance of that in understanding not just the microbiome, but really biology more fundamentally and health more fundamentally. One of the things he talked about, which was really of interest to a lot of people in this group, is this new provider platform that Viome is building, which is why we wanted to invite you.
Yep.
My understanding is that this is a platform that drives home some of those principles from a systems biology perspective that Naveen shared. I think we're all eager to see what you have built, maybe give us a little tour, and I'll ask questions as we go through it. Those of you listening, feel free to type questions into the chat and we can ask those as well. With that, I'll turn it over to you, Momo. Feel free to add anything you'd like to what I just said, but otherwise, looking forward to what you have to share with us.
Okay, great. I apologize that I do not have access to the provider platform, so I cannot do a live presentation unless Guru or Grant joins, but Grant is on vacation, so he will not be joining. Let's get started on what I would like everyone to understand and sort of takeaways. It's a good educational moment without any sort of demonstration. So let's focus on what this is about and what the takeaways are. One takeaway is that I would like everyone to start thinking about the human body, not as a physical object with organs like you were taught in medical school, but think of the human body as a vessel with 100,000 biochemical reactions. About 50,000 are run by our own genes, and 50,000 are run by our microbiome, mostly gut microbiome. So biochemical reactions, not physical objects, not genes. Let's talk about what a biochemical reaction is. A biochemical reaction would be us sensing the presence of food in our body and liver enzymes turning on and producing bile components. That would be several biochemical reactions. Another biochemical reaction would be sending the signal to squirt the contents of bile into the intestines. A bunch of biochemical reactions would be the process of digesting the foods. A bunch of biochemical reactions would be the process of absorbing nutrients from the intestine into the bloodstream. And then a whole cascade of biochemical reactions is what people consider metabolism, converting sugars and fats into energy and converting proteins into making the components of the human body. There are a bunch of biochemical reactions that occur in the liver to detox, and a bunch of biochemical reactions that occur in the kidneys to detox, a bunch of biochemical reactions that occur in the central nervous system that power our emotions, memory, mental health, and cognitive health. So these are just some broad examples of these 100,000 biochemical reactions that make us who we are and that make our body function. Most people are familiar with the term homeostasis. Homeostasis would be a balance of these 100,000 biochemical reactions, meaning we're not producing too much bile, we're not producing too little bile, we're not producing too much cholesterol, we're not producing too little cholesterol, we're not converting too much sugar into energy nor too little. These 100,000 reactions have an optimum range where it's healthy. And these reactions, if they become too fast, so we're doing too much of something, or too slow, meaning we're not getting enough of something, that means we have an imbalance. Let's now translate this into diseases. If we have one of these 100,000 biochemical reactions out of balance, we're not going to have any symptoms whatsoever. It's just such a small part of our physiology. Hundreds of these chemical reactions can break down and they're pre-symptomatic. We don't sense anything.
Yes, it is.
It's saying preparing live stream.
Oh, I don't know what that is. I stopped that. Okay. Let's
All right, let's get back to the programs. The takeaway from what we've discussed so far is that we are powered by biochemical reactions. 50,000 from the human genes and 50,000 from the microbe genes. One takeaway from that is that our microbes are an essential component of our health. They're our best friends. The moment that a child is born, one of the single most important events in that child's life is the acquisition of a healthy, diverse microbiome. That is literally, besides the genes that inherits from their parents, it's the single most important event, because these microbes are going to provide 50% of our physiology to our cells. So they're our best friends, and we need to nourish them and help them and feed them the right foods. So when you're eating a meal, you're not only feeding yourself, you're feeding your gut microbiome. A significant portion of calories in human breast milk are produced by mother to feed the microbiome of the baby. That food, called human milk oligosaccharides, cannot be digested by the baby. The baby doesn't have the genes to utilize that energy. It is specifically made for bifidobacteria
Hmm.
to not only colonize, but to create a healthy environment for physical development, cognitive development, immune development, and mental development. And
Mhm.
so from birth, we're feeding our microbiome, and we need to continue doing that until we die. So
Hmm.
those are some major takeaways from that section. Okay, now let's go to the disease onset. Traditionally, when we say we have a disease, the disease has progressed for years or decades to the point where we can feel it. The body has accrued so much damage that we now can actually feel it. But that damage started years or more likely decades earlier. Going back to those 100,000 reactions, some of these reactions start breaking down in our early age and then more and more break down. And because we're such a complex machine, it's not like one reaction breaks and boom, you have a disease. And let me give you an example. So, when we talk about gut motility, can you tell me if your clinics have any specialties like GI specialties or any specialty?
Some of them do. Everyone is interested in longevity, and so they're all applying kind of
Got
a
it.
approach to preventing disease and optimizing function. But many will come from a foundation in GI, OBGYN, or other specialties.
Okay, I'll give an example. So, gut motility. We all know that there's a distribution of how many times people go to the bathroom. We know that constipation is bad because it's a risk factor for many diseases, specifically Parkinson's, but many others. We know that diarrhea is bad because it's also a risk factor for many things, and especially quality of life. We want people to have regular bowel movements, but bowel movement is not magic. It's not bad luck if you don't have the right one. We now know of 5 factors that control bowel movements. And all 5 of those factors are either solely or heavily contributed by the gut microbiome. So I will just list those quickly. One is bile acids, which is mostly contributed by us. And then we have 4 that are solely or mostly contributed by microbiome, and that is serotonin production, methane production, butyrate production, and hydrogen sulfide production. Those 4 that are produced by the gut microbiome, the only way that you can possibly understand their contribution to gut motility is if you do the Viome gut microbiome test. And the provider and the patient will be given those scores. We have specific molecular scores for serotonin, butyrate, methane, and hydrogen sulfide. Let's break that down. So let's go with hydrogen sulfide, for example. So hydrogen sulfide is a gas. It has unpleasant odor and people make fun of it. But hydrogen sulfide gas is very important for our physiology. And it needs to be made at a certain level. If there's too little production, it's bad for us. If it's too much, it's bad for us. And in one of the ways in which hydrogen sulfide gas controls our physiology is bowel movement. People whose microbiome produces too little hydrogen sulfide gas tend to have constipation. And people whose gut microbiome produces too much hydrogen sulfide gas tend to have diarrhea. Now, display for the fish. Yeah, so I'm waiting for Guru. If Guru can join, I understand that it would be very nice to understand the clinical workflow. Okay, so let's go with the clinical workflow and the dashboard itself. I cannot show you because I don't have access to it. Guru is the one who would, so maybe we can create another session for that. But let's go with the flow. So first of all, I want to say the most important thing, which is that I think that every single person, every patient should have nutritional support, whether they're in their 20s or their 80s, whether they have symptoms of any disease or not. It doesn't matter. They should be provided with nutritional support. I think Viome nutritional support is the most advanced because we measure 10,000 biomarkers associated with chronic diseases that can be modulated with foods. You will see that all in the results and recommendations that are provided. Okay, so that's an important thing. Second thing is you will go to this dashboard and you can order the Viome. It's called Precision Health Pro. is the test. You can register on viomepro.com. So to register as a provider, go to viomepro.com. So viomepro.com. You register there as a physician, and then you can start ordering this test for your patients. What does the test look like? When you order a test, the patient will receive a sample collection kit directly from Viome. The sample collection kit includes sample collection kits for stool, blood, and saliva. They're all collected at home. I can collect all three in 7 minutes, so it's not a huge burden. A normal person, after reading the instructions, can still do it in 15 minutes fairly easily. Saliva is very easy to collect, stool, very easy to collect. Blood is a finger prick blood, and we provide a kit that has these little capillary device that collects just the right amount of blood and puts it into tubes. All samples are preserved at the point of collection at room temperature. There's no refrigeration, there's no freezing, there's no overnight shipping. They simply collect the samples at home and put the samples in the mailbox in the prepaid mailer. They can also drop it off at the post office. Once the samples come here to the lab, I work at the Viome lab and I lead the Viome lab. We have a CLIA certified laboratory for analyzing those three samples. I'm not going to delve too much into the metatranscriptomic details. I think Naveen may have covered that already, and I'm happy to answer questions. But I want you to understand that we are quantifying more than 10,000 biomarkers. More than 10,000 of them we're quantifying in our clinical laboratory. And those 10,000 plus biomarkers are all distilled down into consumerized scores. And these scores would be like a butyrate production score. You do not need to understand all the genes that bacteria use in order to produce butyrate. You do not need to understand how we quantify transcripts using next generation sequencing and how we put those into scores. We do have two very high profile publications that go into the nitty gritty details, including describing exactly how we compute a score and how we validated it in tens of thousands of humans and associated with common diseases. And so all those publications are there. Just go to viome.com/publications and they're all there. So I mentioned, for example, serotonin, bile acids, methane production, butyrate production, and hydrogen sulfide production. Those are 5 of about 70 scores that you and your patient will get. There will be a very strong, clear description of what that score is measuring, how it's related to different conditions. So that's one thing. And then you can drill down and say exactly what foods modulate that score. And oh, here comes Guru. That's great. So he'll be able to go through the dashboard. Hello, Guru. Thank you so much for joining.
Hey, I'm sorry I was not able to join earlier, but I'm hoping that it would still be helpful.
It's absolutely super helpful for you to join. I'm just almost done finishing the background, and they're really curious about the dashboard for providers. And I don't have access to that. So if you can get ready to share with them the dashboard.
All right, let me see if I can do that.
Okay.
Please continue.
So there are these 70 scores that are basically described in such terms that physicians and customers or patients can understand them. They lead to two things. One is they lead to how they're associated with different diseases. So we currently display their association with about 20 chronic diseases, and that number will expand over time as we do more machine learning. And they're also all tied to foods, which foods that person needs to reduce consumption of and which foods they need to increase the consumption of. So everything is tied to actionable items for the providers to see what are the early molecular changes in this person? What are the imbalances associated with what conditions? What is this person at a risk of? And second, what does this person need to do in their kitchen starting today? So it's very actionable. Unless you have questions about that, I think we should now move to Guru.
That would be great. Thank you so much for that introduction. And thank you for joining, Guru. People are super interested to see what the platform looks like. We all believe in the importance of taking a real systems biology view into how we think about health. Hearing Naveen speak about your approach and hearing Momo build on that further has been great. We're really excited to see how all of this comes together in this new Precision Pro platform that you guys have built. Looking forward to seeing that.
All right. Well, first of all, I'm hoping that the audience here, is there an audience right now that's listening in?
There's an audience right now, and then there's going to be a recording that will live on our platform. There's actually a lot of people who watch the recordings because a lot of people aren't able to make the live calls because this is during the workday. There was an issue with the calendar invite. In any case,
Really?
there's going to be a recording, which will be very helpful. And I think there's going to be a lot of interest in watching this.
Okay. I just need one more minute to set this up if you don't mind.
Yeah, no problem. So I guess while you're doing that, I'm happy to respond to what you were saying, Momo. I think we all intuitively understand, at least those of us who practice this type of medicine, how important the microbiome is and all of its influences on our health. The fact that you mentioned about breast milk and oligosaccharides is super interesting. It does make me wonder about, to the extent that we have our own unique microbiome, how the breast milk composition may be, uniquely tailored, not just for the baby to consume, but also for the microbiome as well.
Yeah.
If that is,
They're genetically encoded, and so they are the same. The composition is the same. These cannot be changed. The mother cannot change their genetics, so they are the same. Evolutionarily, we have co-evolved with Bifidobacterium longum to be inherited from the mother. The way the natural human birth occurs is as the baby is coming down the birth canal, it's actually its head is pushing poop through the colon so that the baby and some poop arrive at the exit at the exact same time, and baby gets exposed to poop. That's natural birth. We have basically over the last, let's say, 50 years removed that natural process because poop is gross, right? But that is the natural way. So we inherit from our mom her microbiome, and that microbiome will have this bacterium. Now, an interesting thing is that there's a whole long story here, but let me just summarize it. This bacterium is important, and bifidobacteria in general are important for many things like I outlined, but let me just give you one specific thing. The way that this bacterium was identified is that scientists realized that kids who had this bacterium, their vaccination worked, meaning they were resistant to the diseases. But kids who didn't have this bacterium, the vaccination was not successful. They were still prone to infections. And so then they realized over the last, let's say, 20 years, that it's the butyrate produced by these bacteria that actually stimulates T cells to convert to memory T cells and remember the infection. When a person's immune system gets exposed to a pathogen or a vaccine, their immune system goes up, they clear the pathogen or the vaccine, and then it loses the memory. But if their gut microbiome at that point in time is producing butyrate, butyrate stimulates T cells to become memory T cells. And so that's how this bacterium was discovered. And then it was discovered that it's actually fed by the mother, by the human milk oligosaccharides. And by the way, 20 years after that discovery, now a bunch of baby formulas have HMOs built into them to mimic milk, which for decades we did not have. Then we discovered that this Bifidobacterium longum and other bifidobacteria that feed on HMOs create an environment that stimulates physical, cognitive, mental, and immune system development for the baby. So it's all supposed to happen naturally. Luckily, there's a probiotic, Bifidobacterium longum infantis, available today to purchase. If it was my daughter or son and they didn't have Bifidobacterium longum strain infantis at birth, I would be buying that strain immediately and providing it to my child because it's a fundamental strain for all kinds of development.
Before we dive into that, I think a bunch of people will be interested to hear, is there a specific brand or company that you like for the bifidobacterium?
The original company was called eVivo Biosciences. The company was spun out of UC Davis, whose scientists in the nutrition department identified this bacterium and isolated it. I would only trust that strain because these scientists spent a decade characterizing it. They were super thorough. They published a bunch of work. I will find the company and put it in the chat.
Great.
Okay.
That sounds interesting.
All right.
All right. Well, thank you so much, Momo. We're going to take it away.
Thank you. So I think I have about 20 minutes, so I'm going to try to make it as effective as possible in the given time. So first of all, folks, as Momo probably has described before I joined, there is a lot of insights that are provided to both doctors and patients when you take this test. This test, called Precision Health Pro, has 3 samples in it: a stool sample, a blood sample, and a saliva sample. Once you take the test, you send in the sample, you get a result report, which shows up first in the doctor's portal, and then the doctor gets to review it and then release it to the patient, and the patient sees all the details. Just like our consumer test customers would see all the details, right? You would see all of those details after the doctor reviews it and then releases it. The process for a doctor to get the full report, actually, to even before getting the report, to order a kit for a particular patient, it's pretty standard stuff that you would have probably already done, used, for your other kits, for your other tests that you do. So I'm going to show you a quick summary of it. There's nothing magical about this. The goal is to make it as simple as possible. Here is the screen. I hope you can see it. Yes. Great. So here on the left-hand side, you see the portal. I'm showing here a Dr. Adam Brown who has placed several orders for their patients. Each patient has a test in different statuses. Some of them, the order has just been created, but for some others, the results are pending review. And when you have a pending review, you can click a button called View, in which case you end up seeing the report, which I'm going to show you in just a minute. Here's an example. I have a prototype example here that I can show you. It's not a real patient. And then once you hit this release button, it shows up for that particular patient, and the patient can then see their results and recommendations in this view. So all of that is integrated into the system. The way you create an order is very straightforward. You select a patient from the list and set up the workflow for either the kit being collected at the practice or the kit being delivered to the patient, the payment method either collected at the practice or arranged beforehand. There's a number of different options here. And of course, when you want to set up a particular kit, you would see your kit IDs show up over here, and you can pick one of the kit IDs. And once you create an order, you end up, seeing that person in your order list. We have an integration of our system into a system called Redox, which allows your orders to be taken from any EMR system that you normally use, like Athena Health, Epic, or Cerner, and it can flow through Redox and come back to our portal. So you don't have to actually use this portal. You can use your normal EHR, EMR portal, and your orders from that portal will come back to this portal. And the report for this patient will go back to whatever portal you normally use. This Redox integration will help you do all of those things and make your workflow very simple. Once you get a report for a patient, you get to see a few very important insights right away. In this particular patient, the insights here are that there are 3 diseases that are considered high risk for this person: IBS type D, diarrhea, visceral adiposity, and depression, which happen to be self-reported. And for each of these 3 diseases, there are specific molecular pathways that have been identified as being suboptimal. For example, you have a sulfide gas production pathway that is measured using the Viome Precision Health Pro using the stool sample. If you want the details of exactly what sulfide gas production pathway looks like, you can click on this details button, which gives you the science behind it. So we are trying to look at all the key components of sulfide production. Here you see there's sulfur metabolites, there's sulfur amino acids, there's a number of other components. There's processes like sulfur metabolism, carbon metabolism, amino acid metabolism. These are all accounted for, and these functions are eventually accumulated and compared to a population, a reference population. Based on that comparison, there's a normative assessment that says whether your pathway is normal, meaning in the middle zone, or is it suboptimal, meaning it's less than what is expected and or least of the population reference cohort, or it's higher. If this score is deemed to be not optimal relative to the reference population, then you see the sulfide gas production pathways showing up in one of your underlying suboptimal molecular pathways. And similarly, there's a butyrate production pathway, there's a bile acid metabolism, oxalate, etc. It turns out that these 4 pathways are extremely highly associated with IBS type diarrhea. So we've done the analysis internally within our cohorts of people with IBS type diarrhea, And we've determined that the odds of somebody having IBS type diarrhea is very high when these four pathways are all suboptimal at the same time. So that's why it's shown over here. The methodology for doing that analysis of associating specific pathways being suboptimal to a disease has also been captured here in a methods paper that we can share with people if they're interested. But these are the pathways that show up. Now, similarly, for visceral adiposity, this particular patient has suboptimal pathways for their cellular stress, the cellular senescence, sulfite gas production pathways, which I showed earlier on TMA production pathways. Each of these things has been accumulated, aggregated to show that there's visceral adiposity. For depression, turned out to be a self-reported condition for this particular patient. So what we're doing here is to connect it from the underlying pathways that we're measuring, specifically anxiety-associated pathways and butyrate production pathways that are definitely connected to the self-reported depression. These are the first set of insights that you get. These are our top 3 insights for this particular patient. There's a lot of other insights, but if somebody is interested, the doctors that are looking at various kinds of details, there's a ton of details provided down here, one of which was the sulfite production pathways that I showed you, but there's a lot of other details that are also provided. But let me go back to the most important actions that somebody can take. If IBS type D is considered not to be high risk because of these underlying suboptimal pathways, you can then move on to the actions, the nutrition recommendations, and there's a methodology for the nutrition recommendations that we can also quickly take a look at. Here's the methodology. For all of the foods in our food database in Viome, we have more than 400 foods in the food recommendation database. We go through a glycemic response prediction model. We first determine whether eating a standard serving of any one of these foods results in a spike in the glycemic response. If yes, we consider that to be a minimized food. If it is no, then it goes into an enjoy food bucket. Then we evaluate all of the microbiome pathways. So here we are evaluating dozens of pathways, specifically examples like butyrate production, oxalate, sulfide, and so on. Now, for example, if you have a person who we are looking at spinach for whether it is what type of food recommendation it is for a particular individual, maybe their glycemic response is going to be pretty low for spinach, like it is for most people. But if their oxalate elimination pathway is not good, that recommendation for spinach might drop down to an avoid food because spinach, as you know, is very high in oxalates. If your oxalate elimination pathway is not working well and you still consume a lot of spinach, you can end up getting kidney stones. So we drop that into avoid. We have logic like that for every single one of the foods in our food database. And we go through glycemic response prediction, microbiome pathway analysis, and then we land the food in one of these 4 buckets, which is what you see over here. So when you look at IBS type D, which has been flagged as a risk area for this particular individual, it shows here that you want to emphasize a certain set of foods, you want to avoid a certain other set of foods, and this is based all on the 4 pathways that were considered suboptimal earlier. And then there are also a certain bunch of foods that are considered to be minimized. These are our recommendations based on the analysis that we've done. Similarly, for visceral adiposity, you want to emphasize a certain set of foods, avoid these, and so forth. And for depression, you want to boost your pathways. For that, you want to emphasize these kinds of foods. These are the actions generally provided in the report. Every single pathway is explained. Every decision and method we make is explained. And then the references at the bottom here shows the peer-reviewed publications that we have already published from Viome that shows the data for why we are making the decisions we are for the pathways, for the recommendations. And of course, there's all of our disclaimers and method limitations and so forth. So that's a very quick summary of the report itself. Once you look at the report as a doctor and you say, okay, this looks good, I understand what's going on. Maybe there are some additional things that I need to think about for this particular patient. You're comfortable telling the patient about this, you click the release button. Once you click the release button, the person gets their own recommendation and results delivered to them in an app. We have 2 versions of the app. There's a version of the app on web application method, like through that you can see through a browser. We also have a mobile phone app that you can use, and you get the same information there. This person does not have all the specific issues that we were showing before, but you get this type of information. You can see that for this particular individual, gut and digestive is in the improved mode, and you can also see the longitudinal changes to their gut score over the last 5 tests. You can see that it used to be good, but now it's dropped. So you might want to just say, hey, what's going on? You can look at all the underlying pathways, and turns out for this different patient, their putrescine production pathways, their TMA, their lactic acid pathways are not doing well. And therefore, there could be other disease risks for this particular individual. It's not the same as the previous one. But that's the level of detail that we go into in our reports. I'm happy to discuss any of these details. Sounds like there have been a couple of questions. Should I try to open
Yeah,
those?
go ahead. Yes.
I think those are for me.
Ah, those are for you.
I'm answering them.
And you're answering them. Okay, great. But Anant, does this make sense? Do you have
Yeah.
any questions?
Let me just kind of reflect on a few things. I think one of the things that I appreciate about the approach here is that it is in depth, but it's also understandable. I think it is important to strike a balance between being in depth and comprehensive and being understandable to the physician. Essentially, the model that you guys have put forward here is one in which you say that, hey, the microbiome influences our health in many ways. It looks like you've identified maybe 5 or 7 different key molecular pathways that happen in the microbiome, right? Like sulfide gas production, butyrate production, bile acid metabolism. And what your assay is doing is basically measuring the pathways of the activity of those pathways, and then mapping the activity of those pathways to different disease states. And, I thought what you did with the food was interesting, where it's like you use the pathway interactions or the pathway activity to then make food recommendations, because you overlay, what that type of pathway activity could mean for how a certain food gets produced, right? And so
Yeah.
really that is the model that is comprehensive, but if people can wrap their head around it, they have a sense of what's going on. A lot of times we get reports back and it feels like it's just a complete black box. Most of the
Yeah.
time, when we get even provider facing reports, they look like the patient report, where it's just a bunch of, if you go to home, it looks like this, right? So if I get this, there's a lot of companies that give us scores back like this. It's kind of like, well, what do I
Yep.
tell somebody? It's kind of
Yep.
like, it doesn't tell me like under the hood what actually is going on. What is the reason behind it, right? And
Yeah.
so that's why I appreciate you're actually talking about the pathways that are being altered. And I think as biology continues to mature and as our understanding of different compounds matures, we can start bringing tools in based on our understanding here, right? So if I know that this patient has butyrate production as a problem, well, then we need to target that pathway, right? And there's a range of ways to do that.
Exactly. And we try to capture some of those options that you have. You could provide supplements to directly improve the butyrate content in their gut because it's a short chain fatty acid. You wanted the anti-inflammatory properties and so forth, supplement ingredients, right? However, it's not enough to just have one approach because there's different approaches. Some people may want to have foods that have fibers that then feed the bacteria in their gut that will then improve the natural production of butyrate in their gut. And so that's part of what you see over here. The maitake mushrooms, for example, has inulin, right, which is a complex fiber that is very good for butyrate production in your gut, right? So those kinds of details are captured not only in the methodology that I showed you earlier on, but also in the very quick, simple description of what to do, right? What is going on is this simple dashboard here. And what you have to do is another simple dashboard. If you're sitting down in front of a patient for a few minutes, this is pretty much what you need. Then you can say, hey, look, if you're interested, we can get into the next level and so forth, but let's do this first. Let's see the impact of it, and then we decide what the next steps are.
I love it. Well, I know we're coming up on time here, but I certainly appreciate some of the depth that's gone into this. So is this something that's available to people now if they want to try it out, or where does this?
It is available now. We already have several practices, more than a dozen practices, that are using it now. I'm happy to have anyone in your group, Anant, reach out to us. We can onboard somebody onto the portal that I showed. give them the materials to educate them about what they need to do to understand this, to order this and everything else. And we can get them going today.
Well, if you don't mind me sharing your contact information with
Yes.
anyone who's interested, and I guess whoever the right person is, let me know.
Yeah,
But
you can ask them to send it to me. I'm just giving you my email right here in the notes, in the
Mhm.
chat. So guru@viome.com. If they send me a note, I will forward it to the team that can help onboard anybody from your provider group.
Yeah, no, I think people appreciate that. I know that after Naveen's talk, there were a bunch of people who wanted to check this out, and hopefully there will be some who are interested. I'm certainly interested for myself. So
Amazing.
uh, I appreciate you coming in, Guru, and sharing more about this platform. I appreciate how you guys are trying to bring systems biology based approaches into how we deliver medicine. And I think this is going to be an exciting year for the field and hopefully for you guys as well.
Oh, definitely. We look forward to working with you, Anant, and your team. Any other information, please reach out to me. I'm happy to provide it to you.
Okay, Guru,
Thanks.
take care.
Take care.